If you're struggling with how to write your research articles for publication, this video is going to be exactly what you need. Before I get started, I wanted to let you know that I have a scientific research paper checklist that's going to help walk you through how to do every single part of your scientific paper to submit it to journals. The first thing I would suggest whenever you're working on your introduction is to make sure that you know your research story first before you start writing your introduction. This helps you by letting you know where your research paper is going to go, which is going to give you a much better chance of actually writing a really cohesive introduction that will lead nicely into your research. So for your research introductions, you want to be able to answer 4 specific questions. The very first question that you want to be able to answer is, why is your field important? And so if you've seen other papers that I've done on research paper introductions, pretty much the first thing I'll ever tell you is that the first thing you need to do is tell someone why they should read your paper. And so the first part of that is, why even is your field at large important? Why should anyone care about it? And why should they publish your paper and or read your paper once it's published? And so you can do this really simply by just saying either the impact of your specific field or what your specific field is important for. Once you've clarified why your field is important, you want to answer the second question, which is, what is the background of your field, or what background knowledge does your reader need to know to understand your results? Generally, whenever you're thinking about this, think about the major topics that your paper falls into, and then write the basic background information that someone would need to know about those topics to understand what you're doing in your paper and why that's important within the context of your greater field at large. This is usually going to be a little bit more like a textbook style writing, where you're basically giving them fact after fact, but you want to try and tell it within a story. So you want to make sure that you're leading them from one section of your paper to the next section and not just listing one fact after another. The other really important thing here is to not just include something because you know it. I think a lot of times we go, okay, we know X, Y, and Z, and so we're just going to throw it in and include all of this information. But really, that's not the information that your actual reader needs to know. So you always want to be watching yourself every step through it and being like, okay, is this really something my reader needs to know? Or is it just something that I know and I feel like I want to share it? Once you've answered those 2 questions within your research paper introduction, the third question you want to answer is, what problem is your paper solving? And so this is specifically telling the reader why your specific paper is important to read. They need to know the problem that it's going to solve before they hear how you solve that problem. This very quickly will show anyone the novelty of your paper, because you're specifically identifying why you need this research. It immediately shows the impact and the novelty, which is going to make it so much easier for it to get accepted, and generally easier for someone who's reading it to actually care about it and cite it later on. Different problems that you could be solving is developing a more advantageous method or filling in the gap of knowledge. So maybe we don't know about a certain thing. So you're going to tell us that there's no papers that have really studied X, Y, and Z. And so we set out to study that, and that's what we're going to do in this paper. The fourth question that you need to answer within your introduction is, how have you and others researched this problem? So once you know the problem you're researching, whether it's your specific paper or in this sub area of your field, you want to give people the context for what's been done before. This is generally finding those important papers that led specifically to your research and giving us a little bit of information about what those papers did and what they found, so that we can then understand why you're conducting this study, and we can better understand the results of what you found and their context within the greater scheme of your field at large. And then finally, end out your introduction with a paragraph explaining what you did in your paper and what they're going to be able to learn and read if they continue reading. This is a good way to get people who are reading your paper straight through to keep wanting to read your paper, because you've guided them really nicely from the importance of your field through what they need to know about your field into the problem that your specific paper is solving, and then the context for how it's been looked at before and how you're specifically researching it. So now I want to jump into my own published research paper, and this is the same one I've been going through within this series, but I want to jump through and show you where these different answers to these questions are located within my own introduction. So here's my first published paper. And you can see the first couple sentences here that I have highlighted are telling you why steroid and steroid analysis is important. And so I'm talking about the fact that steroids have a specific role in cellular signaling. As I started to write more and more, I started to refining my first sentence more and more. So more recently, my sentences would say something more like, steroid analysis is important for forensics, medical diagnostic, and sports performance testing, or something like that, which makes it a little bit more clear the importance of it. But even back then, I still started out with the importance of my field at large. And then I immediately jump from steroids importance into steroid analysis importance and talk about how they are potential biomarkers for a variety of diseases and their misuse to enhance performance. So this entire first two sentences is just me stating through why steroids are important as a molecular class, and then why steroid analysis is important, which is what I'm going to be talking about in this paper. So the next things I want to do is go through the background, right? So what are the main topics that I'm talking about in this paper? The first one is steroids, obviously. And then the second one is their analysis, right? So I'm going to be talking about just general analytical chemistry, but then I'm also going to dive deeper into specifically ion mobility, which is the type of analysis that I'm using within this paper. So in the next couple sentences, I'm kind of doing this out of order, but in the next couple sentences, I'm actually talking about the problem that this paper is going to solve. So I'm talking about the difficulty in analyzing steroids. That's exactly what this research was aiming to solve. How can we create a better way to analyze steroids that is faster and can separate out these isomers really well, but very quickly as well? So that's what this whole section is all about, is what are the problems with the current methods, and then I'm going to talk about this new method that we're going to look into and see if it's actually a good way to be able to separate out steroids. The next section that's highlighted in green is all about talking you through what ion mobility is. So now we're going back into that background information. This was specifically sent to a mass spectrometry journal. I didn't dive as deep into steroids because who I'm submitting this paper to isn't as focused on steroids as they are focused on the analytical challenges. So that's why I'm talking about the importance of steroids, but I don't actually give a really long history because my readers aren't going to care as much about steroids specifically. They're going to care more about the analytical challenges that I'm talking about. So you can see here, this is my background information that I'm talking about ion mobility. So I'm talking to them about different types of ion mobility and what ion mobility does. So now that we've hit the importance of my field, the problem that my paper is going to solve, and the background information, the next thing I need to talk about is how am I and others researching this problem. So this is going to go into kind of your literature review section of your introduction, where you're basically going to walk through the major things that other people have done to move this research along. So this next section is again talking about the different literature. So really what I'm doing is specifically saying what each paper is given to the field. I'm covering multiple different papers within this one paragraph because this is an introduction and not a lit review. I don't need to tell you everything that paper did. I just need to tell you what that gave to the field that led to this paper being done. The next section is really a mix of background information coupled with the how people have done this before. So I've made it yellow because the green is the background information section and the red is the how people have researched this before. So I'm specifically coupling in background information about this parameter called collision cross-sections, which is a big part of this paper in studying steroid collision cross-sections. So I'm giving them a bit of background so that they can then understand the context, so the different people who have studied this and what they have found related to it. So that's what's going on in this paragraph. And then, of course, my final paragraph is talking about what am I doing in this study. So I talk about how this study is furthering this body of work. That's a good way to preface that paragraph, is basically saying how your study is contributing, increasing, furthering, whatever, kind of action verb you want to use there, but basically showing how your study is actually enhancing the previous work you're talking about, which is the entire reason why you've included that previous work within your introduction, and then giving a little bit of information about what they're going to see within this study. So I basically talk about my two major findings, that we were able to separate out some steroids using metal adduction, and then we found really good agreement with collision cross-section values that have been previously reported. Those are the two main conclusions that came from this paper, and so that's what I'm including in here as a part of this study. I'm also telling them about the specific steroids I'm looking at, and I have a figure here that goes along with it that you can see in the right-hand panel. And so that is essentially how you would write any introduction. If it seems like it seems pretty easy or simple, it's because it actually is. And it probably has always seemed so difficult because you've never quite known how to do it. In my case, when I first started writing introductions for the very first paper I wrote, not even my first published paper, I didn't know what I was doing. And I remember sitting at a Starbucks for over 8 hours trying to write an introduction, going and analyzing previous papers, and just trying to figure out what I should say. And after that 8 hours, I only ended up writing one paragraph, and even that paragraph I deleted later. If you feel like writing introductions are really hard, it's really because you don't know what you're doing, which is fine. A lot of us don't whenever we're first starting research. And so hopefully this method is going to make it a lot easier for you to write your introductions and seeing how you can actually make it flow. The first thing that I want to talk to you about today is how you're actually organizing your method section. So within your method section, a good rule of thumb is to have one section devoted to one main type of experiment. So this could leave you with sections like materials and solution prep. You could have something like animal care if you're having animals or experiments on the animals. Those are usually can be a single or two separate depending on how involved the care or the experiments are. Then you want to have individual sections for each of the experiments that you're going to be performing in this paper, and then have your own section for analysis. If you're doing mainly one type of analysis through it, you could have a single section for that, or you could include your analysis in each of your different types of experiments if each of those experiments require different analyses. And then in your analysis section, you can basically talk about how you combine the data from these experiments together. So here you can see in my first published paper, I have my experimental section, and I'm just going to show you the sections I have. So the first section I have is my solution preparation, where I talk about where I got my materials from and how I made the solutions. Then I have my ion mobility and mass spectrometry section. So in this case, I'm talking about the actual instruments that I used and their capabilities and how the procedure we used to use them. And then I go into my analysis section. So this is talking about collision cross-section, calibration, and resolution calculation. So this is how I actually analyze my data to show within this paper. And so that's kind of how to section out your method section, is basically devote one subsection to each major step in your experimental process. Once you have your entire method section actually sectioned out, within each section, you want to answer three main questions. The first question is, why did you do something? Now, the first and the third question are kind of optional, but they can really help enhance your paper, where most of us only focus on the second question. Whenever you're asking, why did you do something, this is going to help your reader understand the connection between your hypothesis and why you're actually doing that experiment. So if you're trying to study one thing, you want to say what this experiment is going to do for that thing. Now, on certain things like animal care or even sometimes solution preparation, this part isn't as needed. We know why you have to do that because you need the animals to perform the experiment. The second part is, what does someone need to know to replicate this experiment? If in my paper I'm doing ion mobility and mass spectrometry experiments, I now need to write out exactly what I did that someone else needs to replicate it. So this can include what specific instrument I'm using. Are there modifications to those instruments that I'm using that would be important for someone else to know to replicate it? In my case, if I'm doing eye mobility, I need to share what gas I'm using, and the parameters specifically that I'm using to get this data. That way, if someone's getting different data, might be able to say, okay, these parameters were different, which is why I'm seeing different data. So all of that can be included. Even talking about what software you used for things like data visualization or data analysis can also be really important, because sometimes softwares have slightly different things in them that can result in slightly different results. So think about if you were writing out basically a protocol for someone who's educated in this field to follow, and that's basically what most of your methods section will be. The third question to answer in your methods section is, what are you getting from doing these results? Again, the first and third question are kind of optional here, right? So you could get by with just answering the second question over and over again, and you can have a published paper. And honestly, in my first published paper that I'm about to show you, that's basically what I did. I didn't really talk about why I was doing something or what that was going to lead me to. But if you do these two things, you're going to link from the introduction into your methods, and then you're going to link from your methods into your results, so that people know exactly what that experiment led to which type of results. So when they read your results, they're going to know which experiment that was from. What you will get from doing this experiment could simply be something as, this experiment is going to produce eye mobility and mass spectrometry data for the steroids. Or it's kind of more important whenever you're doing specific types of analyses. So if you're analyzing one experimental group versus another experimental group, what is that data going to tell you in the long run? So now that you know the three main questions, now we're going to jump into my own example. And I'm actually going to critique my own paper, because I think that this method section was written not as great as it could have been. So in my initial thing, the solution preparation, so I immediately just jump into where things are from. This is more like a reference. It doesn't really read very well as a paper. I have how I was making things and stuff like that. So like here, right, when I'm putting in polyalanine was prepared like this, the first thing I would probably do is I would move this stuff and actually have it in a table in my supplemental if I wrote this again, and just had where each thing was from, the compound where each thing was from, and then potentially like a lot number or a product number or something like that for people to be able to easily get their hands on. So that would be the first thing I would do, is remove these two sentences out completely. And then here I would say to calibrate collision cross-section values, polyalanine was prepared. So that now tells you why I'm creating this solution. And then I would say to analyze steroid eye mobility separation and collision cross-section, the steroid solution. So that kind of gives you a little bit more context rather than just saying polyalanine and steroid solutions, because you don't really know why I'm using those two things right now. And then after this, I could say after the solution was prepared, these solutions were then used for mass spectrometry analysis. That's a much better way to connect between those two sections here, instead of just ending it here and then going into this. Obviously, like I said, this was the first paper I wrote. I wrote this like 5 years ago, so obviously I've developed better in my writing since then. So here we have eye mobility spectrometry and mass spectrometry analysis. So again here, all IMS experiments were performed. This is just very dry reading. So what I would probably do here is to analyze the separation of steroids. We used a Waters Synap G2S, right? Or to analyze steroids by IMS-MS, we used. That just gives a little bit more kind of oomph into this and connects it a little bit more to what we're talking about here, instead of just having a series of information here. And then you can see that I'm including how we actually did something. So this is kind of like a protocol. I'm including parameters here that are important for how we did the instrument. And then I talked about how we did the initial data analysis and visualization. So that's kind of the data analysis portion. So I think overall I did pretty well with actually describing what someone else needs to know. But again, I could have, just added a little bit more into this. And then talking about this data was further visualized, we could say something here to help lead into what we got from doing this. So we got a bunch of ion mobility mass spectrometry data, and now we can say that we're specifically going to use this data to understand the collision cross-section and separation. And so again, right now, because of the way that this is actually written out, saying what we're going to get with what I'm doing here actually leads really well into the next paragraph, because obviously here I'm talking specifically about how to do collision cross-section calibration and resolution. So here I say start out with just TWIM's drift time was calculated. So in here I could do to test the accuracy of the CCSs. That was the main reason we were doing this, or to determine how CCSs changed according to metal adduction. That was another reason we were doing it. Something like that to give you a little bit more reason about why did we do CCS values instead of just saying that we're doing CCS values. Come in here. This is pretty good in just telling you what we were doing. Obviously, most of y'all aren't going to be doing this, so I'm not going to go into it in depth. So we have all of this there. And here, the resulting centroid drift times were used with the appropriate to calculate the unknown CCSs. Again, having something where I then said this data was then compared to previous literature values to determine the accuracy of the CCSs, like that's even a part of the methods that I kind of left out here, but that also tells us why we're using this data. And then I go into the calculation of resolution values. So again, for the calculation of resolution values, that kind of starts with why we're doing this. But yeah, I, again, could have probably gone into a little bit more detail. It just makes it like not as dry, right? This isn't really telling much of a story. It's just giving you a bunch of information. And so adding in those two components, tells more of that story. They are optional though. So like if you're literally going to repeat what you said in the next sentence, you obviously don't want to do that because that just makes it repetitive. Instead, sometimes reframing the sentence so it gives a little bit more, reads a little bit more like a story can be better. And then I talk about how it's described. And so this I am talking a little bit more about why we're using it. So that we're using a resolution that actually describes it with two variables. So you can actually tell the difference between two peaks instead of just one, which is typically used in the IMS-MS world. In this video, I'm going to walk you through how to create your results and discussion question, what questions you need to answer within your results and discussion section, and then finally I'm going to walk you through my own published results and discussion section. And in a part of it, I'm going to show you how I answered each of these questions within it. So let's first start with the purpose of your results and discussion section. And the entire reason why you're writing it is really to share the research story that you've created from your experiments. So if you haven't even created a research story yet, this might be what's holding you back from being able to easily write your results and discussion section. So first go and make sure that you've created a story that you can tell with your data so that you're walking people from one piece to another, from one conclusion to another, to really give them the full picture of what you did in your experiments, and you're not just trying to tell them a string of data. For each major figure or conclusion in your paper, you want to have about one section of your results and discussion section dedicated to it. So you want to be able to walk them through exactly what you did, what you found, and why it's important in each of those sections. So now let's talk about the 4 questions that you want to answer in each of those sections that's related to a figure. The first question you want to answer is why you performed an analysis. So whether it's a specific experiment you did or a type of comparison or something like that, you want to tell the reader why you're actually performing it, and this kind of primes them to let them know what they're expecting to get from you performing this analysis. This is somewhat optional. If you're performing a really routine analysis in your field, you don't necessarily need to tell your reader what they're gonna learn from that analysis. You just need to go through how you performed the analysis and what you found instead. So it's a little bit of an optional category, but if you are doing a specific type of comparison, you want to mention what the importance of that is. So if I'm comparing one experimental group to another experimental group, I want to mention that this is going to help me find, biomarkers that are specific for the consumption of caffeine if I have a caffeine and a non-caffeine group or something like that, so that it's a little bit clearer to your reader what they're actually understanding. And this also helps from any misunderstandings due to confounding variables if they're not quite sure what you're expecting to find between the two groups. So once you talk about why you performed an analysis, it's really easy to segue into what you found. So this is really the results part of it. If you have a results and discussion section that are separate, you're going to mainly only focus on just your results here in your results section, where the later two components that we're going to talk about is going to be more your discussion section. When you're talking about what you found, you want to make sure that you're including in your numbers here and your fundamental results. So you want to give evidence to everything you're saying you're finding. So if you're doing more qualitative research, you may be including more quotes in here. If you're doing more quantitative, you want to include your numbers like maximums and averages plus or minus standard deviations. So it gives your reader a really good understanding of the evidence you have that is supporting the conclusions you're trying to make. You also want to make sure that you are mentioning your figures that relate to the data that you're showing in that paper, so that people are easily directed from the text into the figures. The last two questions you want to answer is, why do these findings matter and what do they mean? So if you're writing a results and discussion section that are separate from each other, in the results, you may only have one conclusion sentence that basically goes through what does that data mean without all of the numbers. Then when you get into your discussion, you're going to put that data in the context of your field, so comparing it to other literature, and then saying more about why it matters to your field that this finding was found and what it overall means within the field at large. When you have a combined results and discussion section, you're going to do this all together. So you can move straight from what you found and then directly go into why it matters. So giving the context behind that finding and then concluding with what does this overall mean for your field at large. And those are going to be your main conclusions driving to your overall conclusion of your paper. So those are the 4 main questions you want to answer for every major section or figure within your paper. You're going to answer why you're performing a specific analysis or comparison. Then you're going to go into what you found, and then you're going to answer why does it matter and what does it mean. And so now I want to walk you through my own published results and discussion section. Here's my results section for my first published paper. In analytical chemistry, we write our results and discussion sections as a single section. And so that's what you can see here. So you can see that I have one major heading for each of my major figures. So in this case, I have lithium steroid adduction is going to refer to this figure where I show all the different ATDs for the lithiated adducts. Then I have a sodium and potassium adduct separation. So that relates to this figure here where I'm showing sodium on this side and potassium on this side. My next heading is the resolution of steroid isomer pairs. And so that relates to this figure where I'm showing the resolution as a function of molecular weight of the steroid. Finally, I have the collision cross-section measurement, which you can see here. And that's going to relate to this final panel down here where I'm going to talk about how the collision cross-section changes. So that's how I'm actually sectioning out my results and discussion section. It's fundamentally based on my figures. Now I want to walk you through my last paragraph of my results and discussion section, where I'm actually going to walk you through how I answered each of those 4 questions above. In this first sentence of this paragraph, I'm talking about why this analysis is important. So I'm talking about because of the complex nature of this TWIMS analysis, we need to compare the accuracy of our TWIMS calibrated values to known DTIMS values, which can be calculated and not calibrated. So that's preparing my reader for why we're going through these findings and what they expect to see. What my data is going to tell them is whether the TWIMS analysis is on par with the DTIMS analysis. So now in green, I'm sharing what I found. So we found that monomers had a higher degree of accuracy compared to dimers, and I'm giving the numbers here to show them what that level of accuracy was. In pink here, I'm showing why does this matter. So the trend is basically showing that the calibrant we're using seems to do well for monomers, but doesn't seem to do well for the multimer or dimer species that we're answering. So that's going to be really important if people are wanting to identify these later on. If we can't cross-compare between instruments, then it's going to become problematic. In blue here, I'm talking about what it means. So I'm giving more discussion and context within the field for why this could be happening and what it means for the field going forward. I always end with the conclusion sentence of my general findings. In this case, I'm saying that overall our CCSs were in good agreement. However, the dimers are going to be in higher error compared to the monomers. There are 4 main components you want to have within your research article conclusion. The first main component is going through the importance of your field and the importance of your paper. This is not as necessary if your conclusion is held within your discussion section. However, if you have a separate conclusion section, you want to re-go through why the field overall is important, because there are times when people will just go and read your conclusion section, and having that sentence there gives them a reason to read the rest of the paper because they know why what they're reading is going to be important. The second major component of your conclusions should be about your key findings. So this should be the main findings of your results section. So if you think about maybe 1 sentence per figure in your results section, should be about the right length for showing what your key findings are. In this, you really just want to give a 1-sentence descriptor of the conclusion you came to. You don't need to rehash all of the actual results that you got. The third main component that you need in your conclusion section is how this paper actually moved your field forward. This is really helpful to any reviewers or editors so they know exactly what this paper is bringing to the table. If you watch this video above where I talk about how to write a research abstract, I also talk about including this component in your abstract as well. I think it needs to be in both places so that it really clearly specifies out why you're even writing this paper and why other people should read this paper. Once you've covered what your paper is actually giving to the field, the next thing you want to cover is actually what you think should be done after this paper. So this is the future directions from your paper. You want to let other researchers know what you think should be done next. Were there certain problems within your paper that you think could be solved if you used a different technique? Or did doing this study lead you to have more questions? You should include this in about 1 to 2 sentences at the end of your conclusion to nicely sum up your conclusion with its importance, the key findings, what this paper gave to the field, and then where the field should go after this paper. One bonus component that you might need to include within your conclusion section is the limitations of this paper. The limitations of a paper can kind of fit well in the discussion section, or it can fit in the conclusion section. But if you had major limitations to your study, you want to also mention those within the conclusion section as well. Now I want to show you how I implemented these 4 components within my own conclusion section of my published paper. So here you can see I have my published paper, and I'm going to walk you through the different sentences that held these conclusions within it. So this first sentence here is talking about the importance of TWIMS analysis within the steroid analysis field. I'm talking about how it's a key analytical tool, and the main 2 components related to that is group 1 metal adduction and the formation of multimers. The next section is talking about my key findings. So I talk about how we were able to separate out different steroid isomers. And then I also talk about the importance of the CCSs that we obtained. And so this is a specific parameter that we use, and a lot of my work in this went into making sure that our CCSs were accurate as well. I then add one sentence here showing that TWIMS has been useful in the identification and separation of steroids. That's really what this paper gave to the field, in that I was basically saying that this type of instrument could be used in this type of analyses. Then my final sentence is discussing the future directions or the future work, where I talk about whether this separation would hold true whenever you're actually putting it into a mixtures instead of in a standard solution alone. That is the main project that should come next after this study. If you have multiple branch off points from your paper, you can list multiple different topics that you should cover. I'm going to cover exactly step by step what you need to do to be able to write your abstract for your research articles. Today, I'm going to cover the 6 components that you need in your research abstract and exactly how to go through and do it. Then at the end, we're going to actually show you these 6 components within an example. So the first part of your research abstract should answer, why is your field important? This should be the very first sentence of your abstract. This sentence should be illustrating to the reader why they should read not only your paper, but also care about your field at large. This can be something as simple as saying that your field is important for X, Y, and Z, or illustrating a significance of your field. How many people are impacted by what you're trying to study or trying to solve? Then we go into the second part of your abstract. The second part of your abstract should only be about 1 to 2 sentences detailing what the field you're studying is. So if someone's reading your abstract and you tell them why it's important and then immediately jump into the results, they may not even have enough information to even understand the results you're saying. Giving them 1 to 2 sentences of background can help them actually be more productive in reading your abstracts and understand what your general conclusions are once you get to your results. The third component of your research abstract should be in illustrating the gap or the problem in your field. What is your research paper actually solving, and how is that helping out your field at large? This again should only really be 1 sentence, and it gives context for the person who's considering reading your paper to actually understand the novelty of it. The fourth component of your research article abstract is identifying how you filled in the gap or solved the problem that you illustrated in your third component. This is actually just giving a 1 to 2 sentence description of the methods that you did. These methods are going to help inform the results as you go into those. It also directly helps those reading your abstract to know if your research paper is worth reading for them. Some people are specifically looking for research papers just to figure out what methods they should perform or protocols to use for certain methods. Having these basic methods within your abstract can be really helpful not only for your readers to understand your results, but also for certain readers to know whether they actually want to read your paper or not. Then we jump into the 5th component of your research paper abstract. And this 5th component is outlining the key findings of your research. If you watch my video about how to write research papers for publication, I talk about creating a story. That video will be available above. If you've created that story, when you get to writing your research paper abstract, all you want to do is offer 1 to 2 sentences about the major conclusions that you found within that story. The 6th component to your research paper abstract is talking about how this paper pushed the field forward or built upon the field. This can be directly related to that gap or problem that we talked about above. But in here, really, this is what a conclusion should be. So you're concluding what your paper did, but just saying what it did isn't really useful within an abstract because you already have all of the key findings within your abstract already. So instead, talk about how it pushed things forward. Is it the first paper that did X, Y, or Z? And how is it actually moving your field forward? What is the novelty there in your paper? What is the significance of your paper? This is going to be really helpful, again, for people deciding if they want to read your paper and to be able to have it more easily published, because peer reviewers can see within the abstract itself both what the gap you're trying to solve is, what you did, what your key findings are, and then also how those key findings are pushing the field forward. The final step of your research abstract should be talking about the future directions. What do you think the field should be doing after this paper? What are the direct next steps informed by this paper that will lead this field even more forward? Ultimately, these 7 components are really helpful for you to be able to write your research article abstracts in a way that's going to make it more likely that you're going to get papers accepted and more helpful to your readers in the future. So now I want to jump into a previous research article abstract that I've written and show you really the applicability of these 7 components in an abstract. In this first sentence, I'm actually addressing 2 of the components. I did this a little bit out of order of how I talked about earlier, where the first part of the sentence is talking about why my field is important. My field in this case is steroid analysis. It's important for both the medicine and the forensics field. The second part of the sentence is talking about the problem that I'm attempting to solve. So steroid analyses can be really challenging and complex, and that's what I'm going to attempt to solve in this paper using iMobility. Next, what I'm going to do is give background to the field or talk about what my field is. So in this case, I'm talking about the previous methods that have been used in steroid analysis up to this point and why those are not really advantageous. And then I'm jumping into what I'm specifically using, which is iMobility spectrometry, and being able to apply this and talking about how I'm using it in concert with metal adduction and multimerization. This is the background. It's only a 1-sentence background, but this is the background that gives my reader the ability to understand why I'm doing what I'm doing and being able to understand what I'm doing within the context of my methods and results that I'm going to cover. This next sentence here is actually covering all of my methods for this paper. So in this case, I'm using traveling wave iMobility spectrometry. That's my main method. This makes it really easy for anyone searching for papers to know exactly what my method was and to be able to decide whether that paper falls into what they want to read or not read. I also talk about how I'm looking at steroids, multimers, and metal adducts. These are 3 different parts of my methods that are important for anyone who is going to be reading this, even though you really kind of need to be in my field to fully understand what those things are. And then I'm talking about the fact that this is being done in a mixture looking at isomers, so things that have the same mass, which makes them difficult to separate out by mass spectrometry. So overall, this 1 sentence is just chock full of all the keywords you need to understand in general the methods that I used. In this case, I don't need to talk about solution conditions or anything like that because that's not imperative to the study being performed or to understanding the key results. The next big part of this research paper abstract is all about the key findings that I found. So within my figure outline, I can then go and basically for each section or for each main figure, I can have a half a sentence to 1 sentence explaining what the key findings were. So in the first one, I talk about the main conclusion of my first section, which was all about the formation of heterodimers or heteromultimers. The second sentence, I talk about another big conclusion of my paper, which was talking about the agreement between something called a collision cross-section, which is just a property of a steroid, but talking about that between the mixtures that I'm running and the standards that were previously ran. And then my last sentence in this section is talking about the last main experiment that I ran, which is the last section of the results section of this paper, talking about a super complex mixture and seeing if we still maintain decent separation whenever we're actually combining multiple steroids together. The final sentence of this abstract is talking about the significance of this paper. So I'm talking about how this paper is further demonstrating the potential of iMobility mass spectrometry to provide a rapid separation and analysis for steroids. In this case, I'm not rehashing what my key findings were, but instead I'm saying, based on these key findings, this is how this paper is moving the field forward. This abstract actually didn't include the 7th component, which is talking about future directions. As you can see, it still got published, but I think having that extra sentence probably would have really brought it all together and led the reader into directly what could be talked about next. next. I hope this example gave you an idea of how to actually apply these 7 different components into actually creating a really nice cohesive abstract for publication in your research articles. Again, if you're working on a research paper, please get my free download scientific research paper checklist that's going to help walk you through step-by-step how to complete your entire research paper, and it's really going to increase your chances of acceptance by creating a higher quality paper to be published. I hope you enjoyed this video, and I look forward to seeing you in the next one.